Today's lesson is a dedicated podcast lesson to a former student of mine, Maylin. She asks, "Dear Mr. P, can you please explain the life cycle of a cell and more importantly, the process of mitosis?" So we're going to look at pretty much this life cycle, and to understand the life cycle of a cell, we're also going to look at the concept of, or the process of mitosis. So we've got billions, trillions of cells that make up our bodies, and constantly these cells die, right? Whether we cut ourselves, we scratch, a layer of skin off our bodies, we shower, and cells pretty much, we end up shedding dead cells. But these cells somehow, before they die, need to get replaced. And so that's what we're going to look at with this video. Now, majority of the cell cycle, pretty much, the cell needs to grow, right? So we have our cell. All right. So here's our cell with our cell membrane. We've got our nucleus. And this cell has to pretty much find a way to replicate itself before its time to die comes, right? And dying sounds kind of very, but that's the truth of the matter of when it comes to cells, right? Cells and all living things at one point will die. And so for life to continue, these cells need to find a way to replace themselves. So majority of the cell's life, right, is in a process called interphase. And interphase is pretty much the phase in which the cell is growing and preparing for what we call division. So when the time comes for this cell to divide, that is when we're dealing with mitosis. So during interphase, the cell is preparing for this so-called cell division. Now, cell division involves really the packaging of genetic material, right? So pretty much we want to somehow find a way to replicate DNA, right? And be able to create, ultimately, two identical cells. And please excuse my messy drawing. And that's really what cell division or the life cycle of the cell is going to ultimately end up, where we start off with one mother cell, right, and we form two daughter cells as the final outcome. Now, majority of the cell cycle we're going to look at as almost like a pie chart. So I'm going to draw my pie chart, and this pie chart is going to represent pretty much the entire cycle. And the first stage of this pie chart will represent the first phase, and that's what we call the G1 phase. Right? So the G1 phase is the first growth phase. After that, the cell undergoes a process of synthesizing, right? So this is referred to as the S phase, and we're going to talk a little more about these phases as the video progresses. And this is pretty much where DNA replicates. The cell is going to continue to grow and undergo what we call a G2 phase, where pretty much now the cells are going to prepare itself to undergoing mitosis. And then finally, this last phase is referred to as the M phase. And this is where mitosis takes place. And there are different parts to mitosis, or different stages of mitosis, where we're looking at prophase, we've got metaphase, we've got anaphase, telophase, and cytokinesis. And we're going to look at each one of these stages in just a second. So now, the most important thing about with regards to the cell cycle is to have the ability to, well, look within the nucleus, right? And within the nucleus, we have genetic material. So actually, let me draw this a little lower. So we're going to look at the genetic material, right? Because truth of the matter is, the genetic material and the cell is what needs to be duplicated. So the most important parts that need to be duplicated, and pretty much that coding is encoded within what we call the genetic material. And the genetic material in the cell is referred to as chromatin. And chromatin is not really visible. So you couldn't really see the chromatin in the nucleus of a cell if you're looking at it with a microscope. So what happens during the growth of the cell tries to duplicate this chromatin. So all of a sudden it creates a second strand, which again is so thin we don't really see it, right? And this, it ultimately is really one of the first parts of the DNA replication that it's occurring during this S phase. Right? Now, if we're looking at the cells, right, the cells contain what we call chromosomes. Yet we're referring to the word chromatin. The chromatin is really the chromosomal material, the genetic material that cannot be seen using a microscope. Now, the chromosomes that they will eventually form, right, coil the DNA and the proteins that are responsible for the various things that actually occur within the body. Right? It's pretty much the blueprint that identifies who we are as individuals. Now, in humans, right? In humans, we contain 46 chromosomes, or we could say 46 chromatin before they actually form what we call chromosomes. Now, chromosomes are only visible when the cell is actually dividing, right? So if it's not dividing, this chromatin is not really visible, right? So this chromatin ultimately will condense, right? So all of a sudden, these chromatin, they will start to coil up, right? They will start to coil up. And now here we have what we call a chromosome. But a chromosome, and we're going to label this here in the middle. This here in the middle is referred to as a centromere. Right? And if we look at here, we've got an exact duplicate on both sides. So notice this mirror image that's formed, right? This mirror image separates two sister chromatids. Right? So we call each side sister chromatids. Why? And in fact, I like to call them almost twin sister chromatids. And the reason why I like to call them twin sister chromatids is because the genetic material that is found on one side here of this mirror image of this chromosome is exactly the same identical information on both sides. Right? And so this is what we refer to as chromosomes. And what happens is, by duplicating, right, we start off with a single strand of chromatin. That single strand duplicates, right, to forming the sister chromatids, and eventually they start to coil up to form one actual chromosome that has identical information on both sides. And what happens ultimately at the end of cell division, these sister chromatids will separate and create individual chromatin, such as this here, sorry for the mess, in two completely identical cells. So let's look at interphase, right? So during interphase, pretty much 90% of the cell actually spends itself in this interphase stage, right? As we said, right, we've got the growth, the first growth phase, right, our G1 phase, and that's where during this phase the cell actually produces its organelles, additional organelles, it produces new proteins. And pretty much if the cell is in a favorable condition, if the cell is healthy, the cell is ready to move into what we call this synthesis phase, right? And in the synthesis phase, right, think of the word synthesize, to make. We're trying to make an entire copy of the DNA. So as we said, we start off with one chromatin, right? So during this S phase, it forms that duplicate, right? So both sides are exactly identical to one another. And this is what's found during this S phase, this synthesis phase. And now it's ready to continue. However, think about now this cell here, right, that we've got. And within here, we've got the chromatin. The chromatin will try to duplicate itself. Again, we said that in humans there are 46 of these within the nucleus. So for it to fit an exact duplicate, right? Think of all the parts that are within here. We've got the mitochondria, right? We've got little vacuoles. We've got lysosomes. All these organelles need to find a way to duplicate themselves also, so that they exist in both of the so-called daughter cells. So if we're going to duplicate material that is in the cell needs to grow. That's why the cell will spend most of its time during these growth phases, because the cell has to be bigger. Think of in a home, right? You've got your furniture. If you were to duplicate all the furniture in your home, right, all of a sudden the amount of space that's available to you becomes less. Right? So you're going to need more room, right? You're going to need a bigger house, but which you can't really do unless you tear it down and rebuild. But with cells have the ability to expand itself. So by expanding itself, it has room for the additional or the duplicate genetic material, right? It has room for additional organelles that it's going to make. So the cell is going to undergo, after the synthesis of our DNA replication during the synthesis phase, it's going to continue to grow. We've got the genetic material has already been made a copy. Now the cell is ready to go and grow even further. And then it's getting ready to enter what we call the M phase, which is mitosis. So let's look at the cell cycle in drawing form. So what we're going to have is we're going to have our cell. In the cell, we've got our nucleus, and that's really what we're going to focus on. And another thing that we're actually going to focus on are two parts that are found in the animal cell called the centrioles, and these are going to be very important for cell division. So as we said before, we have the chromatin duplicate, right? This phase, we're in interphase, and interphase is pretty much all three of those stages that we refer to, right? The G1 phase, right? The S phase, and the G2 phase. So that's what we're going to look at first. We're going to look at those two phases. So during interphase, the genetic material. Let's try to draw the genetic material with some different colors, right? So to represent different of the 46 chromosomes. But I'm only going to show two sets of chromosomes, or chromatin, should I say, for this cell division drawing I'm going to show. So again, as we said, in order to make room for this, what's going to happen is the cell is going to have to get bigger. So here is our cell getting bigger. Our nucleus also is getting bigger. Oops, sorry for the mess. And what happens is we now are going to have room for all the additional material that we're forming. So the cell is undergoing this growth and synthesis, right? And this is what we refer to as the first stage of the cell cycle, which pretty much the cell, as we said before, undergoes really 90% of the time in this phase. So ultimately, eventually, what happens, we still can't see this. These are what we call sister, right? These are what we call sister chromatids, right? Or chromatin, should I say. And it's not until we enter into our first phase of mitosis, or the M phase, right, which is immediately following this G2 phase. So what happens is the following. The cell has already gotten bigger, right? So now we're going into the phase called prophase. So during prophase, let's make note of some of the things that are occurring. So we've got our cell's already gotten bigger, so it's not really growing anymore. It's pretty much gotten the size that it needs to get, right? We've got now the nucleus of the cell eventually starts to disintegrate, right? So I'm going to try to keep it consistent with the double membrane, of the nucleus. And the nucleus, right? This is the nucleus, right? The nucleus starts to disintegrate, starts to break down. So it starts to break down. And within here, right, the chromatin that we had that formed a duplicate now will start to condense into what we call sister chromatids. So all of a sudden now, with the microscope, we could see this phase, right? And if you're lucky enough to have I guess slides that will show the different phases of mitosis. So what we have here pretty much are what we call sister chromatids. Right? Another thing that's occurring now during prophase is, well, remember when we talked about there were these centrioles? Well, the centrioles are going to slowly start to move apart. Remember how we had here the centrioles are close together? The centrioles eventually start to move apart, and we're going to see just how important they are here, because what's going to happen is the following. They're going to pretty much appear on different poles, right? So different poles pretty much on each side of the cell. And what's going to ultimately happen is they're going to form these fibers, right? These tubules all the way across, right? These tubules are what are going to connect to the centromere, right, to the centromere, right, of these sister chromatids. So centromere, right? These are sister chromatids, but what is a sister chromatid? So one half is a sister, the other half is a sister. Together, collectively, it is called a chromosome, right? And these chromosomes are where genetic material lies. So what happens now, this is prophase. So now nucleus is disintegrating. The what we call spindle fibers here. Let me write this down, right? We've got spindle fibers, or mitotic, oops, didn't spell that correctly. So we've got spindle fibers, right, that form from the centrioles, as we said. And then eventually the cells undergo the next phase, which is referred to as metaphase. Right? Metaphase. And during metaphase, the following occurs. So let's draw our cell. As we said, we've got centrioles that are at the poles of the cell. The membrane has disintegrated, and what happens now is the chromosomes line up along the middle of the cell, or as I like to refer to and how I remember it, the meta, the half, as the Greek word for half, the meta of the cell. So the halfway part of the cell. And let me draw the second one. So they form pretty much along the middle, right, at the center of the cell. And what happens is, from these centrioles, we have these spindle fibers that attach themselves to the centromere, right? To the centromere, right, of the chromosome, the centromere of the chromosome, which is the center of the chromosome. So we've got the entire chromosome on, right? Remember we said if we look at half of the chromosome, we've got sister chromatids. And this is what we refer to as metaphase. Now, next step, what happens? At this point, these spindle fibers are going to start to pull at these chromosomes. Now, remember, we're going to refer to as, for humans, and as we said, humans, right, they have 46 chromosomes. Right? So imagine 46 of these all in line. Of course, I'm not going to draw all 46, but imagine 46 of them. I'm only going to show 2 of the 46 that are actually, it would be forming 2. So the next phase after metaphase is referred to as anaphase. And let's look at what happens during anaphase. So let's continue by drawing our cell. All right. So we're going to draw our cell. All right, here's again our cell membrane. But what happens now is these centrioles that have the spindle fibers, they start to pull at those chromosomes, right? So these spindle fibers start to pull at them. So imagine you're trying to pull at the centromere, at these chromosomes, and what happens is the sister chromatids break in half, right? So all of a sudden, half of the genetic material, or should I say, remember, as we said, right, they're sister chromatids. Why? Because at this split, if we split this chromosome, this side here is exactly the same, has the same exact genetic material as this side. So what happens is they start to split apart and start to carry the same genetic material to polar ends of the cell, right? So they start to pull them away, and now these are referred to as daughter chromosomes. So these daughter chromosomes start to get pulled apart to opposite poles of the cell. And what happens now, we're getting ready for the final stages of that M phase, the final stages of mitosis. And this is referred to as telophase. And during telophase, watch what's starting to occur. The cell membrane is slowly starting to pinch Right? Here's our cell membrane. They start to pinch. Eventually, what's going to happen is remember the nuclear membrane, how it disappeared? The nuclear membrane is slowly going to start to come together again. Right? But now it's going to start forming on both sides. Right? It's going to start forming on both sides of this an elongated cell that is starting to pinch here. So the cell is starting to pinch. What's happening is we've got now these chromosomes. Right. So we've got the exact duplicate chromosomes, daughter chromosomes, in on each side. Right. And eventually, this is going to pinch together to form two daughter cells. And what happens is also the centromeres kind of come together and they form centromeres for the next time these cells are going to go. So when they pinch together, they pinch here and eventually separate and form two exactly identical cells. Right? And here's our nucleus. Right? Here's our nucleus. And inside now, we are back to having our chromatin again, which is invisible to the, so we won't really be able to see it. It's not really invisible, but we won't be able to see it because they're so thin. And so now, and here are the centrioles. Right? So now what we have, and this actual breaking, or once they've pinched together and have cleaved to form two identical cells, this is what we refer to as cytokinesis. Right? The actual splitting of and forming two identical cells, and these two identical cells are referred to as daughter cells. So both of them have the exact genetic material as the mother cell that we started with over here. Here's that mother cell. And look at the end what we end up forming. Two exact cells that were exactly the same as the mother cell, but now they're referred to as sister cells. All right? So pretty much now, plant cells undergo very similar cell cycle. The only difference in plant cells is during this pinch in plants, right? In plants form pretty much a cell plate that separates the two daughter cells. And in plant cells, there are no centrioles, as you should know, going back to parts of a cell. Right? So ultimately now the cell has undergone pretty much its cell cycle, and now these two daughter cells are going to undergo interphase. And this process will begin all over again. So let's recap. Let's recap here. Cell duplicates its genetic material, gets larger, right? Growth phase, synthesis, right? The synthesis being the duplicating of the chromatin, and then it'll continue to grow and get ready for what we call the M phase. Now, once the cell is ready to go enter the M phase, first M phase or mitosis phase is prophase. Nucleus starts to disintegrate. The chromosome or the chromatin starts to condense, forming sister chromatids. Collectively, they're called chromosomes. Spindle fibers start to form from the centrioles, and they start to separate to different poles. Ultimately, the chromosomes start to move in line at the middle of the cell, and the spindle fibers attach to the centromere of these chromosomes. These spindle fibers will start to pull apart and enter anaphase, where they break apart the identical sister chromatids from one another, right? Or daughter chromosome, and forming these daughter chromosomes, where we're now creating that duplicate genetic material that is going to make their way to forming two completely identical cells. And that pretty much is the cell cycle.